go.
Hi, I'm Erin.
I'm Autumn.
And today we're going to be talking about the enzyme acetylcholinesterase. Acetylcholinesterase breaks down acetylcholine neurotransmitters at cholinergic synapses into acetate and choline products, which terminate synaptic transmission. Its main substrate is acetylcholine, but in its various forms it can bind other substrates as well. The enzyme is a hydrolase, so breakdown occurs in the presence of water. It can be found mostly at neuromuscular junctions, but depending on the variant, it is prevalent in many other body locations. It is one of the most efficient enzymes in the body, with a turnover number of about 14,000 for acetylcholine as the substrate. The most well-known inhibitors of acetylcholinesterase are the organophosphates. They have the ability to bind to a serine residue in the active site, which prevents acetylcholine from binding and being broken down. This has the potential to be very deadly, and drugs affecting acetylcholinesterase are highly regulated and controlled.
The most common mechanism of acetylcholinesterase, as just stated, works by using water to cleave acetylcholine into acetate and choline. In doing so, an OH from the water places the oxygen-carbon bond in acetylcholine, creating choline. A hydrogen atom is attracted to the negatively charged oxygen, creating acetate. This process terminates synaptic transmission by breaking down acetylcholine neurotransmitters through enzymatic degradation. You may have heard of acetylcholinesterase inhibition through its interaction with sarin gas, which was often used by the United States and Syria. Sarin was created in Germany in an attempt to make pesticides. It's odorless, colorless, and very volatile, and bonds with acetylcholinesterase to change its enzymatic structure so that acetylcholine can no longer bind. This means that muscles can no longer relax. It degrades very quickly and has a very short half-life, so it will sometimes be created on the ground with precursor chemicals, and it's an irreversible inhibitor, so it creates a strong covalent bond at the active site of the enzyme, and therefore separation of inhibitor and active site is very slow. Sarin gas reacts with the sarin residue's hydroxyl group in the enzyme to create an ester, making acetylcholine unable to bind. And tissues are not affected, so if you survive the attack, you will most likely recover.
In the study of acetylcholinesterase from 3D structure to function, researchers used acetylcholinesterase from the Pacific ray, the Torpedo californica, to make predictions about the 3D structure and function of a human acetylcholinesterase because of its structural similarity between the two. They do this by first purifying and crystallizing torpedo acetylcholinesterase, then producing recombinant human acetylcholinesterase and expressing their catalytic subunit in Drosophila S2 cells. This was followed by purification using affinity chromatography and separation through the SDS page method under non-reducing conditions. The resulting purified crystals were grown using the hang-drop vapor diffusion method, and protein X-ray and rigid body refinement data was collected from the crystals of the recombinant human acetylcholinesterase.
The gene encoding acetylcholinesterase, the ACH gene, is found on chromosome 7. Splice variants result from alternative splicing of this ACHE gene, and exons 2, 3, and 4 are common to all splice variants and are found in the core of human acetylcholinesterase, where they make up the active site gorge. Isoform T is most commonly found in cholinergic synapses of the brain, muscle, and other tissues, and is the major form of acetylcholinesterase. While isoform H is found in erythrocytes, and isoform R is found in embryonic and tumor cells, and is induced under physiological, chemical, and physical stress.
Acetylcholinesterase is naturally found in all animals and is part of the hydrolase class of enzymes. It is also part of an enzyme family called the carboxylesterase type B family, which act on carboxylic esters. These are part of an even larger protein superfamily called the alpha-beta hydrolases. They get this name based on their folding pattern. Subfamilies of the type B carboxylesterases include the cholinesterases and the neuroligands. Two proteins in particular, neuroligand and thyroglobulin, are among some of the most similar proteins to acetylcholinesterase. Human acetylcholinesterase exhibits 45% homology to rat neuroligin 1 and 28% homology to bovine thyroglobulin. In this figure, the areas of the protein structures that are highlighted in gray exhibit the homology between the sequences of human acetylcholinesterase, rat1 neuroligin, and human thyroglobulin. These areas are most commonly found in the carboxylesterase type B domains, which are conserved among all of the proteins and in the active sites.
The acetylcholinesterase family are alpha-beta hydrolases with 6 distinct motifs. These motifs are unique folding patterns that are conserved with the alpha-beta hydrolase superfamily. Each splice variant has a different structure and therefore will have different domains. For example, carboxylesterase B is one of the domains commonly found in acetylcholinesterase variants, as well as other enzymes in the alpha-beta hydrolase superfamily, while the tryptophan amphiphilic domain is specifically found in isoform T to help stabilize disulfide bonds.
The quaternary structure of acetylcholinesterase is different between its splice variants. However, in the most common form, which is isoform T, which is found in the synapses of the brain and the muscle, they can exist as monomers, dimers, tetramers, and collagen-tailed heterooligomers. The functional acetylcholinesterase species that exists at the cholinergic synapses consists of 4 acetylcholinesterase subunits associated with the collagenous CoQ protein, or the protein-rich membrane protein, prima. 1, 2, or 3 tetramers of acetylcholinesterase can attach to the collagen tail of the CoQ, which links the subunits by disulfide bonds. The proline-rich attachment domain, the PRAD, of the CoQ protein is responsible for the attachment to acetylcholinesterase subunits, where the adjacent cysteine residues form disulfide bridges. The PRAD interacts with the tryptophan amphiphilic tetramerization domain on acetylcholinesterase to form stable tetramers with the CoQ protein, resulting in the quaternary structure that is found most common in the human brain. This picture shows where the tetramers of acetylcholinesterase wrap around the collagenous tail of the CoQ protein and form the collagen-tailed heterooligomer.
The anionic subsite is also known as the choline binding pocket. This site is where the acetylcholine's charged quaternary choline group interacts. There are also peripheral anionic sites which serve as additional sites for binding acetylcholine or other quaternary ligands. These peripheral binding sites are believed to serve as a mechanism for substrate inhibition. When acetylcholine binds to these peripheral anionic sites at high substrate concentration, substrate inhibition is promoted. However, when the substrate concentration is low, the acylation step in the catalytic pathway is accelerated, promoting the breakdown in acetylcholine. In the second subsite, the esteratic subsite, the hydrolytic reaction occurs, creating acetate and choline by breaking an ester bond. Imidazole and hydroxyl side chains of histidine and serine stick out into the subsite to interact with acetylcholine and bind serine to the product acetate until water can liberate the acetate from the subsite. The esteratic site contains the 3 residues of the catalytic triad, histidine, serine, and glutamic acid, the oxyanion hole, which stabilizes the transition state's negative charge, and the acyl pocket, which confers substrate specificity.
The most distinguishing feature of the active site of acetylcholinesterase is the presence of the deep active site gorge. The gorge penetrates about 20 angstroms deep through the enzyme and is lined with aromatic amino acid residues. These residues participate in what's called aromatic guidance of the substrate into the gorge. At the bottom of the gorge lies the catalytic triad, which consists of 3 highly conserved residues of serine at the 200 position, glutamic acid at the 327 position, and histidine at the 440 position. They form a charge relay system, allowing the enzyme to participate in covalent catalysis reactions. The triad is similar to that which is seen in chymotrypsin and other serine proteases, except with opposite-handedness because of the acyl pocket, which confers the substrate specificity as previously stated. There have been investigation about other binding sites for acetylcholine on acetylcholinesterase. Investigation of the binding sites show that it can bind not only at the active site, but at a place near the rim of the gorge called the peripheral anionic site. The site can also bind other quaternary ligands. But for any ligand that is bound at this site, the entry of substrates is blocked and the exit of product from the base of the active site is also blocked. This blockage can cause functional changes for the enzyme, specifically in the case of acetylcholine hydrolysis, where the blocking of the exit of the choline product can cause substrate inhibition. The peripheral anionic site is the yellow of this picture, and you can see how it lines the gorge and how the exit of products would be prevented by the structure of it.
So in conclusion, the alternative splicing at the C-terminus facilitates the association of the acetylcholinesterase with structural proteins, and this means that there are many forms of acetylcholinesterase, and they all have different quaternary structures. The acetylcholinesterase is prevalent in different locations throughout the body, but is most prevalent in neuromuscular junctions. And the active site has two subsites, the anionic and esteric subsites. And organophosphates act as inhibitors to acetylcholinesterase by binding to the enzyme and causing a conformational change so that acetylcholine can no longer bind and be broken down. A novel domain found in human acetylcholinesterase is the WAT domain, which helps form disulfide bonds with the CoQ protein, stabilizing quaternary structure.
Okay, that's the end of our presentation. Thank you.